Hello everyone. My name is Björn von Eyss from the Leibniz Institute on Aging in Jena, Germany, and I'm the lead author of our recently published manuscript in the EMBO Journal entitled A Non-Canonical Repressor Function of JUN Restrains YAP Activity and Liver Cancer Growth. I would like to briefly summarize our results on a few slides and highlight their significance here. The Hippo pathway acts as a tumor suppressor pathway in several tissues. It consists of a conserved kinase cascade whose main function is to restrict the activity of two homologous transcriptional co-activators, YAP and TAZ. When activated, the Hippo pathway induces YAP-TAZ phosphorylation, leading to cytoplasmic retention, thus inactivation of YAP and TAZ. During tumorigenesis, YAP and TAZ become more and more active, and they are able to confer oncogenic properties to cells, aiding in their oncogenic transformation. But surprisingly, the core components of the Hippo pathway are rarely mutated in cancer, arguing that Hippo-independent mechanisms exist to restrain YAP activity, which then are deregulated in cancers. To identify these Hippo-independent mechanisms, we performed a genome-wide CRISPR activator screen in MCF10A cells. We made use of the observation that very strong overexpression of YAP5SA, a mutant that is insensitive to the Hippo pathway, leads to cell death. We then screened for genes which, when overexpressed, could rescue this phenotype, arguing that these genes would act as Hippo-independent suppressors of oncogenic YAP levels. And here we identified the transcription factor JUN as one of the top hits. Since JUN itself is a transcription factor, this suggested to us that JUN directly acts on chromatin to impact on transcriptional regulation of YAP target genes. By gene expression profiling and cut-and-run analysis, we found that two classes of YAP target genes exist. First, there exist JUN unresponsive target genes, and here YAP, together with T transcription factors, tend to sit rather closer to the promoter of these target genes, inducing their expression. For the second class of YAP target genes, the JUN responsive targets, this looks different, because here YAP and T, together with JUN, rather sit at distant enhancers, and this is consistent with previous studies showing that JUN, together with phosphoproteins forming the heterodimeric AP1 complex, synergize with YAP and TEAD in activating their target genes. However, we now found that JUN represses YAP activity. So how do these two things come together? We found that increased YAP levels led to induction of JUN expression, and this was paralleled by JUN-mediated repression of YAP activity. Because high YAP activity leads to JUN induction, it enables enhanced JUN binding at joint targets. And here JUN recruitment was not paralleled by increased FOS binding, but exclusively JUN, most likely as a JUN-JUN homodimer bound to these sites. This JUN complex furthermore brings in N-core co-repressor complexes, leading to a local repression of YAP activity. Thus, JUN is able to buffer potentially oncogenic levels of YAP activity. Consistent with this hypothesis, we found that this buffering system must be inactivated to unleash the full oncogenic potential of YAP and to enable the formation of YAP-dependent hepatocellular carcinoma, short HCC. We tested this in an HCC mouse model where we overexpressed specific oncogenes in mouse livers using a heterodynamic tail vein ejection approach. Here we found that expression of YAP5SA together with the constitutively active AKT kinase led to massive tumor formation after only a few weeks, corroborating YAP's oncogenic role in liver cancer. We then asked the question whether we can restrain YAP's oncogenic program by shifting the equilibrium to the JUN repressor complexes. In order to do that, we did the same experiment, meaning we expressed YAP5SA together with AKT, but co-overexpressed JUN. And here we could observe that barely any tumors or no tumors were formed. Therefore, we proposed that JUN is part of a negative feedback loop that needs to be disrupted to allow the formation of HCCs. In this negative feedback loop, oncogenic levels of YAP lead to induction of JUN expression, promoting the formation of repressive JUN N-core complexes, which would normally buffer high YAP activity. During tumor formation, however, this loop is inactivated so that YAP activity in the tumor increases. In summary, our work identifies an unexpected tumor suppressor's role of JUN proteins based on their ability to dampen the transcriptional activity of oncogenic YAP levels. And we can show that this mechanism can effectively suppress the development of YAP-driven liver cancers.